And without much ado, I just want to start. And he's from the Singularity organization, and I'll let him speak about himself and his work.
Thank you, Harish, for inviting us to come and talk. And it's really great to see so many questions for BioCurious. I donated to the Kickstarter campaign when it started, and even though I just slogged through 6 years of getting a PhD, I'm very happy that it's open, and their biotech lab's open for high school students or anyone, and particularly for entrepreneurs who may be stuck at a company and everything is recorded that they do, the IP that they work on in the lab is owned by the parent company. So I think it's great that you get people coming out in evenings and weekends to work on pet projects. I'm going to give an overview of synthetic biology, and then I'm going to talk specifically about a new incubator program at Singularity University, which I'm the program coordinator for, called Singularity University Startup Launchpad. I'm doing that 20% of my time. The rest of my time, I'm a synthetic biologist at NASA Ames, just down the road. I've been there the last 4 years. So before I start, I wanted to just get a show of hands. If you could put your hand up if you're in biotechnology, if your industry is biotech. Okay, that's about five people or so. Could you put your hand up if you, and I'm not going to ask you to, but put your hand up if you could explain how genes are related to proteins. Okay, that's slightly more than the people who work in biotech, so think about 10, something like that. So that gives me a good idea of where to pitch what I'm going to talk to you about and how much detail to go into the background. So I did my PhD at Brown University. I just graduated, and I was working on extremophiles. That's why I'm at NASA, organisms that live in extreme environments. I'm not going to talk about any of the work that I did at NASA, mainly because I did 6 years of it and I'm really bored of it. But if you want to pick up a poster, particularly if you have children who are into space or biology, there's some posters here and there's some more at the back there about NASA's program in synthetic biology. And I'm happy to answer questions about it at the end. So what is synthetic biology? The hardcore synthetic biologists who founded the field, who came up with the name about 10 years ago, say that they're turning biology into an engineering discipline. And they shun traditional genetic engineers as scientists in engineers' clothing, because if you look at the process that genetic engineers do, it's more copy cutting and pasting. It's more hacking at something rather than an engineering discipline. And we could fight if there are any traditional genetic engineers in the room after the talk. So synthetic biology is trying to turn biology into an engineering discipline because we're not quite there yet. We're not able to control biological systems or cells very easily. But that is the goal of the field. And this is what's driving the field, and I'll come back to this later on. Is that the cost of both synthesizing, that's writing DNA, adding A's, C's, T's, and G's together, and reading DNA, that's sequencing your genome or a bacteria genome to find out what's in it and what it's made of, is falling 5 times faster than the cost of computer power. And I'll come back to that later on in the talk to explain why that's important. Question, yeah?
What's the figure of merit?
What's the figure? The dollar value?
The figure of merit. How do you measure that?
I don't know what.
Days, hours, minutes?
So in terms of the computer power, I don't know.
So the time to sequence the human genome and the cost of what's the basis of this?
So sequencing DNA is a commodity. It's a commodity price now. So if you want to sequence a bacterial genome or a human genome, you can go out and buy it. I got this from a newspaper article that I read earlier in the week. I can't remember the source of it, so I'd have to go back and look at it. But I can show you a chart which has both later on, which has both the cost of sequencing, and you can see the decrease in it, and then the cost of synthesis in terms of short oligonucleotides, and then longer pieces of DNA as well. But you can have a look at that chart in a second. Sure.
Yeah, I was just wondering, how much will it take in terms of time to sequence a human genome, maybe 5 years from today?
5 years ago and now today?
In terms of time?
Sure. I think it's probably down to about a day now that you can sequence a human genome now.
Okay.
And 5 years ago, well, let's say the first human genome, which was about 10 years ago now or so, it took multiple years to do.
So we are down to about a day?
Yeah. It's not the area, I don't work in human
Sorry,
genetics. But
just for numbers.
It was over a decade, right? I mean, the first one was, I mean, they spent billions and billions of dollars.
And they used all these spare cycles and laptops.
Yeah, now it's into the tens of thousands or, yeah, it's a dramatic decrease.
No, I was just wondering what it was today.
Right. Yeah, $5,000 or $10,000 now, I think, if not 1,000, that's the goal.
I know the 1,000 is a goal, but I
Yeah.
don't know what it is today.
So a quick primer in biology for those people who didn't put their hand up to explaining how genes relate to proteins. In every single cell of your body, and there are about 100 trillion cells, there is 1 or 2 copies of your genome, and that contains strings of DNA, A, C's, T's, and G's. And the DNA, when it's read, is converted into a transport molecule, transport of information called RNA, and RNA is then translated into protein. And protein is the good stuff. Protein is things like your hair, keratin, or your skin, pigment in your skin, melanin, or insulin. A lot of things that end in "in" that you've heard about are generally proteins. And they do the stuff. They're physical, or they're colored, or they have taste. They're the actual workhorses of biology. So the cool thing about biology, in terms of synthetic biology, is it's a manufacturing technology. It's a self-replicating machine, and that's John von Neumann who first proposed using self-replicating machines to colonize the moon, which is why I'm particularly passionate about using his quote. So every 20 minutes, a cell can double. And if you leave it for 24 hours, the cell can turn into 1 billion cells. So the power inside of a single cell and the self-replication makes it an extremely powerful manufacturing technology that people want to harness. If you take a look a little bit deeper inside one of those cells, then you'll see a strand of DNA. You wouldn't really be able to see it, but through X-ray crystallography we know it's there. And cells, DNA, the machinery to read that sequence of DNA then converts it eventually into a protein. And each cell can make millions of proteins. So if you think about the self-replication of cells and then the self-replication inside a single cell, it really demonstrates the manufacturing power of a cell. I'm going to put up a short video. To show a little bit more what's going on. So this is a, I'll have to wait until I watch it a bit further. I haven't seen this video in a while, whether it's transcription or translation going on. It's transcription, isn't it?
What's the difference?
So transcription is turning the DNA into RNA, and the secondary molecule RNA. The translation is turning it into a protein. So we'll just watch it for a second, and then I'll remember what it's doing. Okay, so this is transcription. So this is the polymerase, an RNA polymerase, which is binding onto the piece of DNA. And this is graphic, but it's happening in real time. And you'll see it release in a second, and that molecule is now running along that piece of DNA, reading it like a ticker tape. And out the back of it, that yellow strand is a molecule of RNA. So this is happening in real time, but it's a cartoon animation of it. All those things flying around, the yellow things are A's. They would be A's, C's, T's, and G's, but the T's are used, but we don't need to go into that. So this is why you need to eat a lot of protein, or a lot of different things in your diet. Yeah, the question.
Can you do that a little more slowly?
Yeah, so it's a 4-minute video.
Yeah.
So what we'll do is we'll go through it once and then we'll go through it again while I'm talking about
Sure.
the rest of the parts. So there goes in one end the A's, C's, P's and G's and out the other end